Good day everyone, I am here showing my video presentation, reporting and explaining the cell cycle, focusing on mitosis and meiosis and their differences. Also included in this video presentation, the life cycle of a gametic meiosis. So first, let us talk about the cell cycle. Cell cycle is an actively dividing eukaryote cells pass through a series of stages known collectively as the cell cycle. Two gap phases, the G1 and the G2, and an S phase for synthesis, in which the genetic material is duplicated, and an M phase in which mitosis partitions the genetic material and the cell divides. First, the G1 phase. Metabolic changes prepare the cell for division. At a certain point, the restriction point, the cell is committed to division and moves into the S phase. S phase. DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. The G2 phase. Metabolic changes assemble the cytoplasmic materials necessary for mitosis and cytokinesis. The M phase. A nuclear division, mitosis, followed by a cell division or the cytokinesis. The period between mitotic divisions, that is G1, S, and G2, is known as interphase. The cell cycle has stages, but let's focus on the mitosis and meiosis stage. First is the mitosis. Mitosis is a form of eukaryotic cell division that produces two daughter cells with the same genetic component as the parent cell. Chromosomes replicated during the S phase are divided in such a way as to ensure that each daughter cell receives a copy of every chromosome. In actively dividing animal cells, the whole process takes about 1 hour. The replicated chromosomes are attached to a mitotic apparatus that aligns them and then separates the sister chromatids to produce an even partitioning of the genetic material. This separation of the genetic material in a mitotic nuclear division or karyokinesis is followed by a separation of the cell cytoplasm in a cellular division or cytokinesis to produce two daughter cells. In some single-celled organisms, mitosis forms the basis of asexual reproduction. In diploid multicellular organisms, sexual reproduction involves the fusion of two haploid gametes to produce a diploid zygote. Mitotic divisions of the zygote and daughter cells are then responsible for the subsequent growth and development of the organism. In the adult organism, mitosis plays a role in cell replacement, wound healing, and tumor formation. Now that we know the phases of the cell cycle, let's look a little on what happens during mitosis. Mitosis has several stages of its own. Within each stage, there are multiple mini-stages, but we'll focus on the big picture for this lesson. The first is the prophase, where the coating on the nucleus dissolves and frees the DNA, which has condensed into chromosomes. The spindle fibers or strings that will help separate the chromosomes also start to form. In the next step, metaphase, the chromosomes line up at the middle with help from the spindle fibers. After metaphase comes the anaphase, in which the spindle fibers start to reel in the chromosomes to either side of the cell like a fishing rod. In telophase, the large cell starts to pinch into two and the nuclei start to come back together. The last stage is the cytokinesis, where the cells actually split into two. Second stage is the meiosis. It is the form of eukaryotic cell division that produces haploid sex cells or gametes, which contain a single copy of each chromosome, from diploid cells, which contain two copies of each chromosome. The process takes the form of one DNA replication followed by two successive nuclear and cellular divisions, the meiosis I and the meiosis II. As in mitosis, meiosis is preceded by a process of DNA replication that converts each chromosome into two sister chromatids. Meiosis is when homologous chromosomes migrate toward opposite poles of the cell during anaphase 1. Sister chromatids do not separate in anaphase 2. In meiosis, example of it, in meiosis in males. Prophase 1, chromosomes begin to condense. Then, homologous chromosomes pair crossing over occurs a recombinant chromosomes. In metaphase 1, its spindle fibers attach to chromosomes and chromosomes line up in center of cell. Anaphase 1. Chromosomes start to move to opposite ends of cell as spindle fibers shorten. Telophase 1. Chromosomes reach opposite ends, nuclear membrane forms. And the last is the cytokinesis or the final stage, which the cell division occurs. The difference between mitosis and meiosis. Although mitosis and meiosis follow the same basic steps, but they have more differences than similarities. Most of these differences relate back to the fact that although both are needed for cell replication, mitosis and meiosis have different purposes. First, mitosis replaces body cells with identical copies, while meiosis creates genetically different sex cells that will be used to create an entirely new organism. Another difference between mitosis and meiosis is that during mitosis, there is only one cell division, so the cell goes through the steps of prophase, metaphase, anaphase, and telophase once. However, during meiosis, there are only two cell divisions, and the cell goes through each phase twice, so there's prophase 1 and prophase 2. Additionally, because increasing genetic diversity is a goal of meiosis but not mitosis, where all the daughter cells are identical. During prophase in meiosis, a process called recombination, crossing over, occurs. This is when non-sister chromatids of homologous pairs exchange genetic material so that the daughter cells are more genetically diverse from each other. So there are 7 differences between mitosis and meiosis. First is the cell division. Mitosis. In mitosis, a somatic cell divides once. Cytokinesis, the division of the cytoplasm, occurs at the end of telophase. In mitosis, a reproductive cell divides twice. Cytokinesis happens at the end of telophase 1 and telophase 2. Number 2, the daughter cell number. In mitosis and meiosis, in mitosis, there are 2 daughter cells are produced, while in meiosis, there are 4 daughter cells produced. Third is the genetic composition. In mitosis, the resulting daughter cells in mitosis are genetic clones, while in meiosis, the resulting daughter cells contain different combinations of genes. Genetic recombination occurs as a result of the random segregation of homologous chromosomes into different cells and by the process of crossing over, transfer of genes between homologous chromosomes. Fourth is the length of prophase. Mitosis. During the first mitotic stage, known as prophase, chromatin condenses into discrete chromosomes. The nuclear envelope breaks down and spindle fibers form at opposite poles of the cell. A cell spends less time in prophase of mitosis than a cell in prophase 1 of meiosis. While in meiosis, prophase 1 consists of 5 stages and lasts longer than prophase of mitosis. The 5 stages of meiotic prophase 1 are liptotin, zygotin, pachytin, diplotin, and diakinesis. These 5 stages do not occur in mitosis. In genetic recombination and crossing over take place during prophase 1. Fifth is the tetrad formation. In mitosis, tetrad formation does not occur, while in meiosis, in prophase 1, pairs of homologous chromosomes line up closely together, forming what is called a tetrad. A tetrad consists of 4 chromatids, 2 sets of sister chromatids. Sixth is the chromosome alignment in metaphase. Mitosis. In mitosis, sister chromatids duplicated the chromosome, comprised of 2 identical chromosomes connected at the centromere. Seventh is the chromosome separation. Mitosis. During anaphase, sister chromatids separate and begin migrating centromere first toward opposite poles of the cell. A separated sister chromatid becomes known as daughter chromosome and is considered a full chromosome. While in meiosis, homologous chromosomes migrate toward opposite poles of the cell during anaphase 1. The sister chromatids do not separate in anaphase 1. Below is a mitosis and meiosis Venn diagram that summarizes all the key mitosis versus meiosis similarities and differences. On the left side of the diagram, you can see the key features of mitosis. On the right are the key features of meiosis. And where the 2 circles overlap is where their similarities are listed. So here it is. Included in this video presentation, the life cycle of a gametic meiosis. The gametic life cycle is the reproductive cycle found in animals and some protistans. The term gametic refers to the fact that gametes are the result of meiosis. During the gametic life cycle, a reproductive cell produces haploid gametes, sex cells such as egg and sperm, that combine to produce a zygote. The zygote grows by cell division and cell elongation to produce a multicellular diploid individual. In the gametic life cycle, the gametes are the only haploid stage found in the life cycle. The gametes, where it is the egg and the sperm, are the only haploid cells produced. So that's all for my short video presentation that explains about the cell cycle, focusing on the stages of mitosis and meiosis and their differences, and also includes the life cycle of a gametic meiosis. Thanks for watching. God bless.